So problem I have with this class is that we try to teach you a whole lot of stuff. And you got DNA structure, RNA structure, you got DNA replication, you got transcription, you got translation, you got protein stable folding, you got protein activity, then you move into membranes and cells and signal transduction, and just so much stuff. There's not enough time to treat it in any real detail. So you guys are just learning the surface material of each one of these sections, and that in large way is unfortunate because you should, as a BMS student or as a budding scientist or a medical professional, should have a thorough understanding of this process. I'm not given enough time to do that. But what you, I hope, appreciate is that there's lots of tRNAs. Each one has a specific sequence located at something called the anticodon. And each one of these tRNAs carries a very specific amino acid. You've looked at the genetic code, 64 different anticodons, 3 of them encode stop codons. That means 61 of them code for amino acids. And there's only 20 amino acids, so there's a lot of redundancy built in, and you've thought about that. I know that you've thought about that part. So the tRNA that comes in would be a perfect match for the codon in the messenger RNA, and that's indicated by this structure here, and you recognize it as a messenger RNA because it's got a poly A tail and it's got a 5' cap, so we know we're in a eukaryotic system. Okay. So the tRNA comes into the vacant A site, and that's where all the magic happens. The one piece of magic that actually happens, that is peptidyl transfer, occurs between two tRNAs that are sitting in the A and the P site. And all this long-winded story is just meant to tell you that it's the transfer here of the amino acid or acids that are in the P site that are transferred over onto the A site. It's not the other way around as you might intuitively imagine. Okay, and so that amino acid, which was in our case the initiating methionine, is now transferred onto this one, which we'll call amino acid number 2. So I'll label this 2 to be consistent. So the amino acid 1 is transferred onto amino acid 2, and we looked at the mechanism of that transfer last time at the very end of the class. We'll look at it again at the end of this set of slides so that we understand it. That leaves an empty tRNA sitting in the P site, and during the process of that transfer, these 2 tRNAs slide, and they slide over into the E and P site. So now the empty tRNA, the one that's shed its amino acid, if you will, is sitting in the E site, and the tRNA that now has grown by 1 amino acid is sitting in the P site. That tRNA sitting in the E site then leaves, leaving us back in this situation where one site is occupied, and it's the P site, leaving us again a vacant A site. So this site here that's open now would be the A site, ready for another tRNA carrying another amino acid, and then of course the RNA has been moved down at the same time, and now it's looking at the next codon in line. That process continues. I guess that's what I'm meant to pick up from. We have peptidyl transfer making a 1 amino acid polypeptide into a 2 amino acid peptide, and then a new tRNA comes in. That growing peptide is then transferred onto it, giving us now a 3 amino acid peptide. You notice that the one that's now the last amino acid in line is the one that just came in to the ribosome. Those shift over, one leaves, leaving a vacant A site, and again, a new amino acid comes, the peptide itself is transferred onto that amino acid, and so on. Okay? Over and over and over again, until the stop codon is reached. Okay? And there's 3 stop codons that are recognized, and they don't bind a tRNA at all. In fact, they bind a protein. Looks a lot like a tRNA, so it kind of mimics a tRNA, but it slips into that vacant A site and then kicks everything out. It's a really cool story that you should think about sometime. Okay, so that's the basic story of protein synthesis. What you can